Okay, Bio47, this is actually the second part of Lab21a. So we finished the vessels below the heart. So we looked at the arteries and veins below the heart and down into the lower extremities. And now I wanted to finish up the lab looking at a teeny little bit of fetal circulation and then blood cells. So, or blood and then blood cells. So for fetal circulation, one of the things that's really important is to recognize that obviously the fetus is inside its mother and is relying on a constant supply of blood from the mother to get nutrients and respiratory gases, and then also to relieve itself of waste products. Now, that is done through a pregnancy organ that's only present during pregnancy, and that's the placenta. And then the exchange between the placenta and the baby will be the umbilical vessels. So remember, arteries are always going away from the heart and veins are always going toward the heart. So if we're looking at the fetal vessels that are coming off the placenta, those being the umbilical artery and vein, you have to remember, if you're carrying blood to the fetal heart, that the umbilical veins are going to be carrying blood rich in oxygen from the mother, rich in nutrients. The mother obviously will have balanced the nutrient load with the maternal liver, also would have removed waste products with the maternal kidney. And so what's traveling in the umbilical vein toward the baby's heart is going to be blood that is hopefully ready for use by organs and tissues. What is traveling away from the baby heart and then back to the placenta would be the used blood from organs and tissues. And so that would be the umbilical artery. So if you're looking at color coding, you have to remember that in the baby, the red is going to be the umbilical vein. That's why you see it labeled there. And the blue is going to be the umbilical artery. So the artery, of course, this is pressurized blood, but it's coming from the baby and it's going back to the mother because the baby's heart is actually beating. And then same thing for the mother, the umbilical vein is actually going to be a pressure vessel, even though it's called a vein, but it's coming off that placenta and traveling to the baby's heart. The other thing we have to remember is that the baby's lungs are not being used at this point. So obviously the baby's inside the mother's womb and it's not able to actually exchange air and respiratory gases through the lung. And the liver doesn't need to actually function because the mother's done all the work in balancing the nutrient load. And so the fetal liver doesn't really have a workload at this point. So I have here fetal lungs, kidney, and liver are not fully functional in the fetus. The fetus relies on the mother's organs to regulate fetal blood. And the organ of pregnancy that functions as the exchange surfaces for fetal and maternal blood is going to be the placenta. All right, and then the umbilical vein delivers blood to the fetus, and the umbilical arteries are moving blood away from the fetus and back toward the mother. All right, so with this, now we're going to look at the heart and what's happening in the heart. And this is really all about the lungs not working. At this point, the baby's not breathing, so the lungs are collapsed at this point. They're filled with a lot of water, and so there's a lot of resistance to blood flowing into the lungs. So that's part of the reason that we're going to see that the lungs don't get really well perfused with blood. You get a little bit of perfusion, but not very much. So as blood comes into the lungs, remember that blood is coming from the mother and it already has a high level of oxygen in it. So you don't have to travel through the pulmonary circuit. So when you're coming in, here we have the inferior vena cava. This is going to be oxygenated blood from those placental vessels. It's going to go into that right atrium. It will mix with some deoxygenated blood coming from the baby, but this blood is still fairly oxygenated relative. It's then going to travel into the right ventricle and into the pulmonary artery. This pulmonary artery normally would go to the lungs, but remember, this fetal blood doesn't need to go to the lungs. And so what we're going to do is we're going to have a bypass, and there's actually two. The first here is between the right atrium and the left atrium, and the second is between the pulmonary trunk and the aorta. So highlighted in yellow here are the two shunts that really help make sure that most of the blood that comes into the baby is bypassing the pulmonary circuit and going directly into the systemic circuit. So this first connection or shunt is called the foramen ovale, right? Foramen, remember we use this term a lot with the skull, means whole, and ovale tells you about the shape of it. This allows us really nicely oxygenated blood coming from the mother to travel from that right atrium directly into the left atrium. So we're going to shunt it across. Once it goes in the left atrium, that left atrium will allow blood to come into the left ventricle. The left ventricle pushes that out into the aorta. Now, not all of the blood gets shunted across. We do have some that goes into the right ventricle. From here, if it goes into the pulmonary trunk, we have one more shunt. This is called the ductus arteriosus because we're going between one artery and another. So it's a duct or a passage between the pulmonary artery and the aortic artery. So it's called the ductus arteriosus. And so again, we have one more opportunity to bypass the lung circulation and go directly into the systemic. Okay? So the ductus arteriosus connects the aorta with the pulmonary artery, further shunting away from the lungs into that systemic circulation through the aorta. Now, these two spaces will naturally close when a fetus is born. Most of them close immediately, not functionally or not physically closed, but functionally closed. As soon as a baby takes a breath, the resistance in the lungs goes down, and blood will then be able to start perfusing lung tissue. It's one of the reasons it's really important that those babies take that first initial breath so that they can start perfusing their lung tissue. And then the pressure changes within the right atrium and right ventricle, left atrium and left ventricle change. And at that point, the pressure in the pulmonary circuit drops. And so that kind of sucks blood into this pulmonary circulation. That now means that there's a greater pressure here. It means there's more blood going into the systemic circulation. So we're going to see these shunts start to close. All right, so the one other shunt that we find, so we have two shunts that exist within the heart and one shunt that exists within the liver. Remember, the baby's blood doesn't need oxygen and the baby's blood doesn't need to have balanced nutrients, so it doesn't need to go through the liver. So here, remember, you have an umbilical vein carrying in this wonderfully balanced, nutrient-rich and oxygen-loaded blood. That vein, is the umbilical vein, is going to come in, it's going to run right along the inside of the abdomen all the way up to the liver. If you remember, we had that falciform ligament, and then there's the round ligament, which is a thickening in it. And that then is going to pass directly into the ductus venosus. The ductus venosus is a shunt that shunts essentially past the liver and directly into the vena cava. So we're not going to really go into the liver. We'll have a small amount that travels into the liver. The liver's not functioning at this point. We do need nutrients to support liver growth, but not metabolism. So most of that blood will go directly into the vena cava through this duct that goes from vein to vein, and we call this the ductus venosus. So we'll go vein, umbilical vein to vena cava. So we have a ductus venosus, we have a ductus arteriosus, and then a foramen ovale. And all of these should close when the baby's born. Excuse me. Depending on which one, the ductus venosus should close within the first month or so. The foramen ovale can take a little bit longer to physically close. And the ductus arteriosus, usually it's about a week or so. All right. Next is looking at blood. And so when we look at blood, obviously this completes our whole cardiovascular component in the lab. But when we look at blood, blood is a connective tissue. So remember way back when we looked at tissues, we have epithelial tissue, connective tissue, muscle, and nervous. And I told you when we did the connective tissue is we spent time on the connective tissue propers, and then the supportive connective tissues. And I told you eventually we would get to the fluid. And so this is a little bit about our fluid connective tissues. So blood is a fluid connective tissue. Lymph will be our second. We'll talk about that in lecture on, this week, today. I haven't done that lecture yet. All right, so what we're looking at is the inside of a blood vessel. So these are all endothelial cells. This is on the arterial side, and you can see red blood cells and white blood cells, and these little tiny things called platelets. If we look at blood, again, it's a fluid connective tissue. It has two major components. Just like any connective tissue, remember you have your matrix, and then you have the cells that live in that matrix. For fluid connective tissues, the matrix is called plasma. So if you compare that to, for example, cartilage or bone, remember for cartilage, we had the chondroitin sulfate matrix, kind of that bouncy stuff, and you had those little pockets, the lacunae, and the cells lived in the lacunae. Here we have a fluid matrix. It's mainly water and dissolved constituents within that water. So you can have water with electrolytes, soluble proteins, soluble nutrients. You're going to have soluble proteins carrying maybe some steroid hormones or soluble hormones and so on and so forth. So that's all in the fluid compartment. Oh, and dissolved respiratory gases. That makes up, and there's a gender bias. Women have more fluid than do men. But you have approximately 55% of your blood volume is going to be the plasma, which is that fluid component or the plasma matrix, right? And then here, this is a little sample of blood. You can see this kind of straw or amber colored fluid kind of floating to the surface. Cells are kind of heavy, so they're going to kind of sediment with time to the bottom of a test tube. And the majority of these cells we're going to find out are going to be red blood cells, and so that's why it has this red color. There are also going to be white blood cells and platelets in here. The cellular component makes up about 45%. So again, males are going to have more cells and a little bit less fluid. Females are going to have more fluid and a little bit less cells. So there's a little bit of a gender bias, and that's because of bone density and muscle mass in males is greater. And so men have to have more red cells. All right, so if we're looking at the fluid component, again, this is just the watery matrix of blood. We're going to have 99% of it being water and about 1% of it being soluble organic. This is supposed to be molecules and minerals. So those things are soluble. So the soluble component, again, a short list would be things like your electrolytes, calcium, sodium, magnesium, potassium, soluble proteins like your clotting factors and cellular messengers, cytokines and things like that. You're also going to have soluble respiratory gases, so your carbon dioxide, which can be solubilized, a little bit of oxygen solubilized, and then nutrients, so glucose, amino acids, fatty acids, and glycerol. So that's all the soluble component, but the majority is just pure water. Okay? When someone's talking about what's the difference between plasma and serum, serum is just really plasma without clotting factors. All right, so now we're going to look at the cellular component. So we'll look at the cells. When we talk about cells pretty loosely because a cell has to have a nucleus to be a true cell. And we're going to find that the only cells that actually have nuclei are going to be our white blood cells once they're circulating. Red blood cells and platelets actually don't have nuclei once they're circulating. Now, they are cells when they're developing within the bone marrow, but once they start to circulate, they're going to actually not have nuclei. So oftentimes we're going to use this term corpuscle to describe them. All right, so all of our blood cells are formed in our bone marrow. So that's where they begin life. In the bone marrow, we have what's called a hemopoietic cell. Hemo meaning blood, and poiesis means the birth of. So we have a standard hemopoietic cell with, under the guidance of hormones and cell messengers, will develop into either red blood cells or white blood cells or platelets. And then these will escape the sinusoids within the bone marrow. They'll go through little fenestrae and enter into fenestrae, very similar to what we see in the glomerulus, and they enter into circulation. So these are quick little drawings. These are red blood cells. These are white blood cells and platelets. These are the three cellular components that exist within blood. All right, so we're going to talk first about our red blood cells. So I don't necessarily care so much you know exact numbers, but just to kind of give ballpark. Again, on average, the majority of your cells are going to be these red blood cells. Their fancy name are erythrocytes. So erythrocytes, there you go. Erythrocytes are the red blood cells. Their main goal and role in the body is exchange of respiratory gases. One of the things, if you take physiology, you'll find is that oxygen is extremely insoluble. And if we were to measure the amount of oxygen contained in plasma in a deciliter of blood, you have about 0.3 mils of oxygen that's able to be dissolved versus 20 mils of oxygen carried on red blood cells. So there's a tremendous difference when blood has blood cells in it versus no blood cells in the sense of its oxygen carrying capacity. So the majority of the oxygen is actually carried on these blood cells. Get a little bit of carbon dioxide. Most of the carbon dioxide actually gets dissolved within the plasma. When you look at the red cells, again, they make up about 99%. So if you had a cubic millimeter of blood, you'd have about 5 million red cells. If you take physio, and we actually do a face-to-face lab, that's one of the really fun labs we get to do, is you get to actually measure and count the number of red cells you have in a cubic millimeter. Their lifespan is about 120 days, and one of the reasons that they are able to last for so long without a nucleus is they really aren't doing very much. They just kind of circulate around in your body. The enzymes that exist within them and the proteins that bind hemoglobin are actually made before they escape the bone marrow, and then they just kind of circulate around until they start to break down. And then we'll see the immune system kind of recirculates and recycles them. They're made at an astounding rate. They estimate that we make about 3 million new red cells every second. And that makes sense. We consider you have about 5.5 liters of blood, and within a cubic millimeter, you have about 5 million of them. So there's a tremendous turnover of these cells. All right, but main thing is erythrocytes' main role is respiratory gas exchange. That's their primary job. They're tremendous in their numbers, so they're the most common blood cell. They make up 99% of a sample, which is why blood looks red. They're essentially pretty high in concentration when we look at number of cells within, or percentage of cells within the blood. All right, so they are not round. I think a lot of us think of cells as always being round, but these guys are little flat, kind of biconcave discs. And so the reason they're flat and biconcave is because when we talk about anatomy, we always talk about folding, and hopefully you've recognized a theme. Anytime we have folding, it's really to increase surface area. And that's what we're doing here, is we're increasing the surface area for gas exchange. So you have this nice surface area where you reduce the internal volume or space, and you increase the surface area to volume. When you look at a single red cell, which I keep doing that. So here, for example, you can see we've reduced the volume inside and increased the surface area to volume ratio by having these nice bisymmetrical sort of indents in them. The other thing is that when they travel through the little tiny capillaries, the capillaries are going to be about the diameter of one of these red cells. Now, their approximate size is about 7 micrometers. And so when they're traveling through, we don't want them to bounce into each other or bounce off of each other, because that creates turbulence. And so what they do is they form these little stacks, which are called rouleaus, and they travel like a little train through the little capillaries, and makes nice smooth blood flow and reduces the likelihood of blood clots and things like that. So they're called biconcave discs. This is to increase the surface area. And they're flat, which makes them stack efficiently, which reduces turbulent blood. So they kind of flow through in we call a rouleau. All right, inside of a single red cell, they estimate there are about 280 million of the hemoglobin molecules per red cell, and each hemoglobin molecule can hold up to 4 oxygen molecules. So you have about 5.5 million red cells per cubic millimeter, and each blood cell can contain about 280 million hemoglobin, and each hemoglobin can carry up to 4 oxygen molecules. So that's a tremendous amount of oxygen that's traveling in our blood. We have a very high metabolism. So this is looking at a hemoglobin molecule. It's made up of 4 different polypeptide chains that have what we call a porphyrin ring in the center. So this is the heme group, which then has an iron. And iron is really it's really what gets oxidized. When iron is oxidized, it's rust. Essentially, that's oxidized iron, and it has this nice red glow to it, which is why blood, when it's oxygenated, looks more red. Iron that is devoid of oxygen has a much more blue color, or a cyanotic color. So the more deoxygenated the blood is, the bluer it gets. Now, venous blood, like I mentioned, is not actually blue. And that's because venous blood is about 70% oxygenated. We always have a fairly good oxygen reserve, which is why you can hold your breath up to about 4 minutes without dying. It's because you have a fair amount of oxygen still left in that venous blood. All right, so I have here the heme is really this porphyrin ring. It's a chemical structure that holds an iron, and it binds reversibly oxygen. One of the things you'll spend time looking at in physio is that binding and unbinding relationship relative to the blood's acidity, which is kind of cool. So the globin part, we talk about hemoglobin. Globin is the actual polypeptide chain. And so when we have hemoglobin that is oxygenated, we call it oxyhemoglobin. Again, it has that redder color to it. So this is oxygenated blood, bright red. And then deoxygenated blood, they always have it blue in coloration, especially when we look at diagrams and things like that. But if you look here, this is deoxygenated blood. If you had venipuncture ever done and they're drawing blood, they're going to draw it from one of the superficial veins in your forearm or antecubitus, so usually the median cubital vein or the basilic. And if you see that blood, it should be a nice kind of Merlot or dark kind of reddish color. So that's because this blood is not actually without oxygen. It just has less oxygen, 70% oxygenated versus about 99% oxygenated. All right, so this is a little quick video on erythrocyte oxygen transport.
All cells need oxygen. It is the essential fuel which is necessary to enable cells to stay alive and to carry out their various activities. Bringing oxygen to the cells requires the uptake of oxygen from the air in the lungs, its transportation in the blood, and its delivery to cells all over the body. The first step is the taking up of oxygen by blood flowing through fine capillaries in the walls of the lungs' air sacs, or alveoli. The oxygen molecules change from their state as a gas freely circulating in the air, dissolving into a solution in the plasma within the capillaries of the alveoli. Once in the solution of the blood, 98% of this dissolved oxygen is taken up by passing red cells, leaving just 2% remaining in the physical solution unattached. Red cells are particularly well suited to transporting oxygen because they contain a special oxygen-binding protein known as hemoglobin. Each molecule of hemoglobin itself contains 4 molecules of heme, an iron-containing pigment, which binds oxygen loosely and reversibly. Hemoglobin that is fully saturated with oxygen is bright red and is called oxyhemoglobin. On the other hand, hemoglobin that is not saturated with oxygen is purplish blue in color and is called deoxyhemoglobin. It is heme which makes it possible for the red cells to pick up oxygen dissolved in the blood, transport it combined with hemoglobin, and release it back into the blood as oxygen in solution, ready for delivery to the various cells of the body. Hemoglobin gives up its oxygen as red cells travel through capillaries in tissues where there is a low content or partial pressure of oxygen. The partial pressure of oxygen represents the level of dissolved oxygen in plasma. As oxygen is released and again is carried in solution, the partial pressure of oxygen in the capillaries becomes greater than the partial pressure of oxygen in the surrounding tissues. This causes oxygen to move out of the capillaries into the tissues and to finally reach the cells. This graph, the oxygen dissociation curve of hemoglobin, shows why hemoglobin is particularly suited to its role in transporting oxygen. The oxygen dissociation curve demonstrates the relationship between the oxygen carried in combination with hemoglobin, the O2 saturation, and the partial pressure of oxygen in the arterial blood. The sharp upstroke and the flat plateau illustrate how oxygen is released to the tissues over a wide range of conditions. Its shape means that although the partial pressure of oxygen in the blood returning from the lungs and being pumped out by the arteries may be reduced to only 50% of the normal value, say due to lung disease or high altitude, hemoglobin will still be 85% saturated with oxygen.
Okay, so we don't really need to know about the partial pressure. That's more of what we would do with physio. But at this point, we want to look at the white blood cells. Now, let me just mention this, that they're in fact white. They do not have pigment, but there's 5 of them. And in order to figure out who's who, histologists will actually add dyes. And that's where they get their fancy coloring. In fact, their nomenclature or their naming refers to the colors that result when you add stain to them. So you'll hear names like eosinophil and basophil and neutrophil. Phil means like, and baso, neutro, and eosin refer to the stains. So when we look at them under a microscope, you're going to see that they have all these fancy colors to them, but they're called white blood cells. And sometimes students have a problem reconciling that, but realize that when we see them through a microscope, the only way we actually can see them is if they're stained. Otherwise, they don't have any pigment. They look like these little ghosts that kind of circulate around. So white blood cells or leukocytes are essentially our internal sort of army. They're our immunity. They are what recognizes what are our self cells and what are foreign cells or debris, those things that need to be gotten rid of. So there are 5 different white cells, and each one has a slightly different role or function, but collectively, they're able to do an amazing job for our immune system. So they'll do things like recognize what's foreign. They'll be able to actively fight. So they'll be eating and grabbing and digesting different cells. They can eat foreign substances, so particulates that are put off by these foreign pathogens. They'll be able to detoxify, so they'll be able to secrete oxidases that can oxidize and detoxify things. They can increase blood flow to where there's an area of infection. And probably most importantly, is they can remember an infection. So that's actually now in the middle of this coronavirus, they're talking about vaccines. That's what they're trying to figure out is what is it about this virus that our body's going to be able to remember. And so if we were able to expose a person to some part of the virus that the immune system can recognize and then remember it. That's what's really important, is being able to remember it, so that if we get exposed to the living coronavirus, our immune system can more effectively fight it off. So remembering an infection or a previous exposure is really important. So these are just really nice scanning electron micrographs. So in this one, you have sort of a pile of red cells and then a single white cell. This is very typical because remember, red cells make up 99% of all the blood cells. That means that white cells and platelets make up about 1%. So they are far less numerous. In the same cubic millimeter that you might have found 5 million red cells, you might find 10 to 12,000 white cells, so much smaller numbers, unless you are fighting off an active infection. This down here is looking at a white cell that's in yellow here. They're coloring it. And the blue things are actually bacteria. And you can see it wrapping around and grabbing. And it'll actually pull that in, endocytose it, and then eat it. Okay. All right, so there are 5 different white cells, and the way we classify them is according to their appearance, not what they're doing. So when we add stain to them, we notice that of the 5 white cells, 3 of them look like they have little grains of color, and so they call these the granulocytes. And 2 of them didn't have those little grains of color, stain, that we added. So those are the 2 major classes. So we have granulocytes and what we call agranulocytes or non-granulocytes. So there are 3 granulocytes. They're called eosinophils, basophils, and neutrophils. And what they're talking about are the color of the granules. Eosin is a red stain that gets added, and so the eosinophils like eosin. The blue staining granules are called basophils because the basic stain, these cells essentially bind really well to that basic stain, so they have blue granules. And neutrophils have granules, but they don't seem to have a preference for either red or blue, and so they call these neutro or neutrophils. All right, and then the 2 agranulocytes or non-granulocytes don't have any granules in the cytoplasm, and so we'll talk about those more specifically as individuals. Okay, so here are the 3 granulocytes. First of all, when I'm looking through a slide and I'm trying to figure out what I'm looking at, remember you should always have a red cell in your frame of view because they make up 99% of all the cells. So always use your red cell as a way to measure, right? So you have always this little microscopic ruler. Red cells are pretty standard at about 7 micrometers, 7.5 micrometers. So when we look at granulocytes range between 12 to 14. So I like to think of it as if I took a erythrocyte or red cell, I could fit two of them approximately across the distance of one of my granulocytes. And that's true for all three of them. So their size is about 2 times bigger than a red cell. That's first and foremost because the agranulocytes are going to be different in size. So all of the granulocytes should be about 2 times bigger than a red cell. And then when you look at them, they should look like grainy. The cytoplasm has these little punctate distributions of stain. Again, eosin is a red stain. So you can see here the cytoplasm is sort of stippled with this red stain. What's in these little vesicles is actually peroxidases, and the red stain adheres really well to those peroxidases. The basophils have blue staining granules. So inside of here is going to be histamine, and histamine is going to stain really well with the basic blue stain. So we have basophils, we have inosinophils, and then the neutrophils, you can see granules, but they don't have either a red or blue preference, and so they call these neutrophils. All right, so when we look again at a slide, so these are actual histological slides, remember they're stained. We always stain with a basic stain, and what always stains blue are nuclei. Chromosomes really like, chromatin really likes the blue stain. So you should always see blue if you have a nucleus. So notice all of these red cells here, they kind of look like targets, and that's because the center is a little bit narrower or thinner than the outside edge. And so there's more hemoglobin in the outside ring. So that's why they kind of look like little targets. And then we have our white blood cell. I notice again, it's not white, it's colored, and that's because we add stain. Remember, the red blood cells don't have a nuclei, so that's why we don't see anything purple in here, but the white cell has a nuclei. We're going to find that the first of the granulocytes we look at is called an eosinophil. We can see this sort of granular, kind of punctate, sort of textured cytoplasm, and it's red. Right? And then you have bilobed nuclei. So the nuclei always are bilobed in eosinophils. So when you look at a cell and you say, oh my gosh, it's twice as big as a red cell, it kind of looks reddish, and it has a bilobed nuclei, that's going to put you in that group of eosinophils. These guys make up about 2 to 4% of a blood sample when you're looking, 2 to 4% of the white blood cells in a blood sample in a healthy individual. If you have an active allergic reaction to something, you typically see those people who have allergies are going to have more eosinophils because the body is trying to detoxify sort of some foreign agent. Basophils are very, very special. We don't have a whole lot of them, so if you were to count 100 cells, you may or may not find one of them, which is crazy. So 100 white cells. These guys are also twice as big as a red cell, so you kind of fit 2 red cells across the diameter of this cell. And it just looks bumpy with sort of blue dots all over. Because the granules stain the same color as the nucleus, it's often difficult to see the shape of the nucleus, but it is actually sort of a indented nucleus. So it has a little bit of an indent in it, kind of like a kidney bean. And you just see these blue granules all over. These guys are containing histamine, and they bind really well with our basic stain, which is why we call them basophils. And these guys increase vascular permeability, and they dilate the blood vessels. So if you have an infection, this helps allow blood cells to get out and into the area of an infection. All right, neutrophils are by far the most common. So these guys can make upwards to 70% of our white cells. So when you're looking at a slide, you're going to see lots of red cells and just a ton of what we call neutrophils. These are also called polymorphonuclear cells because their nuclei is multiple lobes. So poly means many, morph means shapes. They have all these cool shapes to their nuclei. You can tell they kind of have a stippling to the cytoplasm, but it doesn't seem really red or really blue. And they're about twice as big as a red cell. So we have 1, 2, 3 neutrophils here. This video is a video of a neutrophil cruising around, and what their job is, you can see this little bacteria, is to find something that's foreign. They don't even know what it is, so they just know it's not supposed to be there. They eat it, like this guy did, and then these vesicles, which don't stain really well here, have digestive enzymes, and it basically degrades it inside, which is kind of cool. So these are basically your Hoover vacuum of your immune system. They cruise around and they eat cellular debris, old red blood cells, pathogens, all kinds of stuff that shouldn't be in the plasma. All right, so those are your neutrophils. So those are the 3 granulocytes. Then we move to the agranulocytes, and size is really important. So again, you have your red cell as your ruler, and what you're going to find is that the granulocytes, or the agranulocytes, sorry, make up the biggest and the smallest of our white blood cells. So monocytes are the largest of the white cells. So we typically look at the ability to maybe fit 1, 2, 2.5 to 3 red cells in here. So I have they're 2 to 3 times larger than a red cell. Their nucleus is this nice big biconcave. So you have this big indentation, huge kidney bean shape to it. So again, they should be about 2 to 3 times bigger, have a big indentation in the nucleus. These guys are also similar to the neutrophils. They're going to be phagocytic, so they're going to be big eating cells. And these make up about 2 to 8% of a white blood cell count. All right, and then the smallest cells, but also the second most numerous of our white blood cells are our lymphocytes, and these guys are super important. Lymphocytes can be about the size of a red blood cell or almost the size of one of our granulocytes, depending on if they're active. But if they're inactive and they're not making any of our immune products, they're going to be quiescent and about the size of a red cell. So that's what we see here. This is pretty quiet. This is the cytoplasm. Doesn't have a lot of cytoplasm, which means there's not a lot of sort of cellular machinery. Has a nice solid nucleus. There's a lot of genetic material that it can ramp up what it's doing. But its job is going to be twofold. It can divide and specialize and become what we call T lymphocytes or B lymphocytes. And then the T lymphocytes can develop into cytotoxic T cells or killer cells. And the B cells essentially divide and become antibody factories. So they're the ones that are going to remember a previous infection. So super important. So when we look at our lymphocytes, these are what we call cell-mediated immunity. They have a very specific target. They recognize one particular sort of invader, and then they start to mount an immune response. And so that'll be sort of collecting in large numbers. So they're going to divide, and increase in number. And then they're going to secrete antibodies, which will help to tag and flag foreign agents. And then they'll initiate an army, an infantry that'll go out and bind and start to kill these invaders. So these are our lymphocytes, and they are the smallest of the white cells, but they make up the second most populous group of our white cells. They're about 20 to 30%. All right, and then last but not least are thrombocytes. These are cell fragments, and what was here was they are fragments of a cell called a megakaryocyte. And so a megakaryocyte is going to develop in the bone marrow, and then it fractures into these small pieces. And each one has an aggregate of clotting proteins and factors that will be used in creating blood clots. So these are all little platelets. All right, so the last little thing on this would be just to be able to recognize these. So if I gave you a plate like this with different cells, being able to recognize who's who. So first of all, if we look at this plate here, we can see our red cells around the circumference, and we see 2 white cells. Okay, the 2 white cells here have these cool shaped nuclei. They're about twice as big as a red cell. And the nucleus, the cytoplasm has some sort of coloring to it, but it's not particularly one color or the other. And so in this plate, these would be 2 neutrophils. Same thing down here. This is another neutrophil. So these are my polymorphonuclear cells. Again, you have multiple shaped nuclei. The cells are about twice as big as a red cell. And although it does look kind of stippled, I can't really tell what color, right? So neutrophils, lots of them. You typically will see them side by side. This guy right here is, we call an eosinophil. So when we say red and blue, it's more like red-ish or red-er. So it often is not like a bright red, especially because you're adding both red and blue stain to the same cell. It's just which one stains more. So I like to use my nucleus as a gauge to color. But in this case, you can see it does have little punctate distributions of color here. So these are my granules. They are redder than the nucleus, and the nucleus is bilobed. And the cell is twice as big as a red cell. So if the cell is twice as big as a red cell, has a bilobe nucleus and redder staining granules, I have an eosinophil. Now, if I look down here, I'm also looking at, so this would be another eosinophil. Again, I look at my bilobe nucleus. Again, the granules are redder. So it's not a huge bright red color. They're just redder than the nucleus. So this would be another eosinophil. This is a basophil. Notice the dark, dark blue, and I can't really see the nucleus. So I have my basophil, again, twice as big as a red cell, dark staining blue granules, and I really have a hard time seeing the nucleus. I'm going to go down to these guys here. These are all lymphocytes. So when you look at a lymphocyte, you're looking at a cell that typically has a round, solid nucleus, right? And has a little halo of cytoplasm. There's so little in this case that this cell is just quiet, right? Waiting to be sort of initiated by an infection. Same thing here, you see just a tiny bit of halo of cytoplasm. So these are all kind of quiet lymphocytes. They're about the size of a red cell. You can see that here, dark solid nuclei. This guy here we're not going to really talk about, but this is an activated lymphocyte, which is called plasma cells. This one's making antibodies. So you can see the lymphocyte can grow into this guy. So this is an antibody factor. So they call those plasma cells. All right, and then last but not least are these guys, and these are our monocytes. Each one has a slightly different shaped nucleus, but you're looking at these kind of solid nuclei, have this little indentation. Sometimes you can't see it depending on the orientation, but big, big, big cells. You can fit usually 2 to 3 red cells across the diameter of it, okay? All right, so let's test our knowledge. Let's go through these. We'll start with this guy up here. So if you were trying to figure out who this was, look at the size. So how many red cells can you fit across it? Well, you can fit about 2. It does look like it has dots in its cytoplasm, and I have this cool shaped nuclei. So this has to be my, oh darn it, there you go, neutrophil. I kind of already gave that one away, but oh well, neutrophil. And then upwards to 70%. So this is going to be your most common or most numerous, just huge eating cell. I have over here my eosinophil, which I already labeled, unfortunately, but you have that nice sort of reddish stippled cytoplasm. I have my bilobe nucleus, and again, it's about twice as big as a red cell. All right, so down here, yay, this one, I see dark blue. It's kind of hard to see the nucleus. Twice as big as a red cell, so this should be my basophil, right? Less than 1%. So these are very rare. And then over here, nice solid round nuclei. It's about the same size as a red cell. I do see a nice little halo of cytoplasm, so that's my lymphocyte. Right, and then last but not least, I have the big guy here, well, relative big, and I have a nucleus that doesn't seem to have a lot of bumps to it or vesicles, stipules to it, solid nucleus, and so that's going to be my monocyte. So in the sense of numbers, most white cells are going to be neutrophils, followed by your lymphocytes, and then monocytes and eosinophils are about the same in a sample, and then you very rarely see basophils. You get super excited if you see a basophil. All right, and then this is just a blood sample like you might have seen in lab. You can see these 2 cells here, they're already labeled, so these are neutrophils. And then we have a lymphocyte. Notice it's just solid with really no cytoplasm, and it has some platelets. It's kind of nice to look at, and lots of red blood cells. This one is nice because it's not labeled, but you can see A, that would be our red cells, right? So those are erythrocytes. B, notice about twice as big as a red cell, have this cool nuclei. Cytoplasm doesn't have a red or blue color to it, so those should be your neutrophils. Down here, I see my bilobe nucleus. It's twice as big as a red cell. It has red staining granules, so that should be my eosinophil. And last but not least, solid nuclei, little bit of cytoplasm, so this should be my lymphocyte. Okay, and then I think this is the last one. This is just showing us a nice neutrophil there. And then these are big monocytes over here. Sometimes they get squished, but these are big monocytes. And then this little guy is a lymphocyte. I have my lymphocytes and then my monocytes.